much. Can you all see my presentation? Do you see it? Okay.
Yes, perfectly.
Well, thanks for giving me a chance to present our joint work on the genetic population structure of pike around the island of Rügen based on pool sequencing. This is work, the genetic part of it has been done in my lab together with Stefan Denmoser, but the development of the project and the nice field work and a lot of the sampling has of course been done by the group of Robert Arlinghaus, which I want to acknowledge here. So let's get into the pike. What's interesting about pike around Rügen? They are an important natural resource and they're potentially an ecological key species that is heavily used but matters to the ecosystem. So there is a need for informed management, and that's why we want to learn about the population structure. And then of course, there are open questions on pike biology that have already been touched in the previous presentations. And the previous presentations have also nicely introduced some general patterns about the population genetics of pike. First off, the diversity of pike overall is very low. We have only a small number of species worldwide. And usually the species that have been studied across a larger range have a very low genetic diversity. However, some studies, including one that we have done on our own on German pike, that one is the one that produced the tree you can see in the image, has generated evidence for population divergence at smaller scales, and that can be found even, as the previous talk said, in the Baltic seas, but even within single lakes like Plöner See in Germany. So we're interested in understanding why populations diverge. And in order to do that, it is evident that we need high resolution studies because of the overall low levels of genetic divergence. And with respect to Baltic pike, we have some indication that brackish water populations may not be heavily affected by stocking. That was just touched in the discussion on the last talk. We know that population subdivision is partially explained by salt content. There are several studies, and Joanna's talk just presented a very nice example of that. But we also know evidence for this from the Rügen area, which is our focus study area. And then we know from other studies that pike can be anadromous. So all of these pieces of evidence taken together give us very clear evidence that these things are going on, but they do not give us a genome-wide perspective. And that's really where our study complements the ones we have just seen. We do have a genome-wide perspective and can offer some resolution because of that. So let's go back to the study area, the island or archipelago of many small islands around Rügen. You can see sampling sites written out in different colors. In black, we have the bottom areas, which are mostly brackish water. We do see small streams and rivers written out in white that enter the bottom waters in the study area. And of course, we would first like to know, do we have the same patterns of population subdivision that have been described from further east in the Baltic? So do we have brackish and freshwater residents, and do we have anadromy? And of course, are there genetic traits causing this population subdivision? So what are the traits, the actual genes that matter? And in order to address this question, we have collected samples of 50 individuals from each of the sampling sites in here and performed pooled sequencing of complete genomes. So for that purpose, we have extracted DNA of individual pike and mixed those DNA samples from one sampling site into a pool. That pool was chopped into pieces and sequenced to great depth on an Illumina sequencer. And for the analysis, well, we took the reads we obtained from the different populations and mapped those reads against the available and nicely annotated Pike genome that's pretty good quality already, which is nice. And we scanned along the complete genome to identify nucleotides that are polymorphic, so-called SNPs. And with those SNPs, we can then generate the FST values we have previously seen in the other talks. So this is a part of a, well, basically this is a graph of the chromosomes, one besides the other, a so-called Manhattan plot. On the y-axis, you see for every genomic position, for every SNP that we found in the data, we see the FST value plotted. And this graph is really based on millions of pairwise comparisons. So what can we do with this? We can, of course, study the overall population structure as we've seen it in the previous talks and address the questions which lineages exist. On the other hand, this genome-wide perspective really puts us into a position where we can filter out those genomic regions that show maximal divergence, and we can link that with the underlying genes to identify genes of interest. And for the purpose of follow-up studies, we can identify highly diagnostic SNPs for a genotyping assay. And we can use that to type only a handful of SNPs to assign individuals to lineages. And I have highlighted some examples of such genomic areas of high divergence in red, where you can see that there's really a number of SNPs in a very small genomic regions that have an outstanding genetic divergence in such a population comparison. So let's go a little bit into the result. Here you can just see small images of different comparisons among fresh and brackish water pike. And the two images on the top left are comparisons between two river or four river populations, independent comparisons. And you can see that the FST values, the average is going to be like some level on that axis. The axes are all the same scales, and the FST values, the average is written out in the images, so it's in the same order of magnitude that we have seen before. If you go to the right side of those images, you can see the FST values are lower for three independent comparisons of brackish water populations, all as expected from the previous talk. And then in the bottom left, we can see a comparison between a freshwater and a brackish water population, and there the FST is again higher. So low divergence among brackish water pike, high divergence among isolated freshwater populations. We can take the complete data and then generate a neighbor joining tree to visualize how the sampling sites are related to one another. And what you can see here, the colors on the map match what you see as colors on the tree. You can see the black cluster. That's a group of 4 sampling sites that we find in the highest salinity, and they seem to cluster together in the genome-wide genetic analysis. Likewise, we find a group of populations that clusters best. They're surrounded by the blue circle, and those are definite freshwater populations that we know about. And then there's ongoing studies, which I'm going to get into following this slide, that suggest we have anadromous populations entering the Serobach and Neuendorfer Hechtgraben for spawning. And genetically, those seem to be intermediate between the other 2 clusters. A very interesting and nice study that complements our genetic study is a movement study on 300 radio-tagged pike that is conducted by Félicie Delem and Robert Arlinghaus. And they have really marked individual fish and are able to track their positions in the whole study area. And here you see the movement patterns of Brackish-water pike. This is an example for those fish that stay in the brackish bottom waters for the complete year, and they do not enter freshwater at all. So that's evidence that these brackish water populations are indeed brackish water populations. And there's other individuals that were found to enter Zerobach for spawning. And after the spawning, and this is what we see in this image, those individuals move out of the small stream and also live in the bottom area. So that's extremely nice evidence that there's anadromy going on. And those fish, for our genetic analysis, they have of course been sampled in Zerobach during the spawning time. And finally, just to round it off, there is evidence for freshwater pike, and these freshwater pike have been tracked for the whole season, and they seem to stay in the freshwater systems. Here, for example, you see the Peene River, and they spent the entire year in these rivers. So that's the complementary study for which we have actual movement data that show what the fish do, and that shows that our genetic tree, the overall population structure, which I've just shown, matches these behavioral patterns. So if we go deeper into the genetic analysis, we may pull out candidate genetic regions, and I will just pull out one, and I can tell you that there's not too many of those, but this is one that came out with a very interesting pattern for us. For example, because of the annotation of the genome, we know which SNP is associated with genes. And here you can see differentiation at the STAT5A gene that is known from literature to be acting as a growth hormone and to be involved in osmoregulation. And if we compare any of the freshwater populations and zoom in into the FST plot, the Manhattan plot on the top left, you can see that there's nothing conspicuous between the two red lines, and that's the area where the gene is. The same applies if we compare two brackish water populations. In the bottom left, we see nothing conspicuous between those two red lines. However, if we take different freshwater and different brackish water populations in independent comparisons, we see repeatedly that there is a peak that is one of the stronger peaks in the complete data we have. And this peak indicates that there's genetic differentiation associated with this locus. And that's, of course, well, in the absence of functional genetic studies, it's a smoking gun, but at least it's a peak of strong divergence, and literature says it's involved in a trait relevant to dealing with salt water. I want to conclude with this and move on to my perspective. We have a few candidate traits that may explain population structure. I should rather say adaptive population structure. Those are the genes that count. And we have a few SNPs with maximal divergence. With this, we have developed a genotyping assay that allows us for very efficient genotyping using amplicon sequencing on a next generation sequencer. And we can, with the approach, we can type a large number of fish that can be then individually assigned to the genetic groups that we have found. And that permits us to now really dig in and perform high resolution studies on pike biology. So the next thing that we have actually planned for the coming year is to genotype an impressive set of 3,800 individuals collected by Robert's team to find association of these genetic traits with life history at the individual level, which is something we have not yet done because we've done the pool sequencing approach to first find the loci, and now we'll go back to the individual loci. With this, I want to conclude. I want to thank the many local fishers and who have contributed to the sampling of the fish, and of the authorities for support and funding. And I want to thank you for your attention and be happy to answer questions now. Thank you.